welcome, and thanks for inviting me. Yeah, I've got the topic clinical importance of HIV sanctuary sites, reservoirs, and latency. Well, I would like to postulate that HIV has proven itself to be the ultimate survivor contestant. And despite patients being treated on HAART for many years and achieving undetectable HIV viral loads, viremia will still occur in patients within a week after stopping the treatment in such patients. HIV is able to outlast the onslaught of HIV, and this is mainly because of latent HIV viral reservoirs and also sanctuary sites. Now, currently, the only hope to actually cure those that are already infected with HIV basically lies in attempting to eradicate these HIV reservoirs and sanctuary sites. Now, I'm not sure if all of you are aware of this, but currently the International AIDS Conference is underway in Washington, D.C. And there's quite a few VIPs attending the conference, including President Bill Clinton, Hillary Clinton as well, our own South African Deputy President Kgalema Motlanthe, as well as Bill Gates, Sir Elton John, etc. Now, one of the important topics that they are actually raising at the conference is the mechanism of HIV latency. And hopefully by the end of this talk, you will understand why it is so important that we do actually understand what the mechanisms of latency are. So this is just an outline of the talk. We're quickly going to touch on the different aspects of HIV latency. We'll look at what the mechanisms are, what are the clinical implications of HIV latency, which ultimately is why we're looking at this, and also the therapeutic approaches to latent HIV. Okay, so first off, to start with the aspects of HIV latency. Now, first off, there are persistent viral infections can basically be divided into two different categories. The first one is then latent infection, and this implies a persistent infection within a cell, and there's an absence of viral gene or transcription in these cells. And there are certain conditions that might activate the cell in order to initiate viral transcription again. Then you have productive persistent infections, and here basically your infected cell is not actually killed by the virus, so viral replication still occurs. And typically in these cells, the virus will exit the cell by means of exocytosis or budding. Okay, so this brings us to the concept of viral decay. Okay, now the principle here is that plasma virions that you find will have a half-life of about 6 hours. And the level of HIV RNA in the plasma will correlate strongly with the half-lives of the actual cells that are infected with the HIV and that produce the HIV. So if you measure HIV RNA levels in plasma and you plot it against time after the initiation of HAART, the plasma viremia will basically go through 4 different phases of decay, which corresponds then to the half-lives of the different populations of HIV-infected cells. Okay, so this is the basic principle. And this will then, in other words, represent the composition of the HIV viral reservoir. So directly, and this is directly related to the possibility and to the time needed for antiretroviral regimens that are currently in use to actually cure the infection. Okay, so if we put that into a picture, firstly, the phase 1. Phase 1 is basically a cell population that has a half-life of only 1 to 2 days, and presumably these are your active CD4 T cells that are infected. The phase 2 is basically 1 or more cell populations that have a half-life of about 1 to 4 weeks. And these are, for example, infected macrophages, dendritic cells, or partially activated CD4 T cells. Then you get to phase 3, which has a half-life of 39 weeks. And then phase 4, which is a constant phase. And as you can appreciate, there's no appreciable decline which is caused by the activation of resting memory CD4 T cells that actually start to produce virus, and possibly also infected hematopoietic progenitor cells. So if we look at, again, just a different representation of this with the different time time lapses here. And as you can see, also in your third and your fourth phases, you will also have some viral blips that occurred in this period. Okay, which brings us then to persistent viremia during HART. Okay, so we know that HART can actually interrupt the HIV life cycle, and then it suppresses your viremia to levels that are lower than the detectable limits of our conventional assays. So this typically will be, at the moment, about 50 copies per milliliter. But despite dramatic improvements in the patient's well-being, the eradication has not been accomplished as a result of using these antiretroviral agents. And we know that if we stop HART, that patients will have a detectable HIV viral load again. So we know also that with our new ultra-sensitive viral load assays, that there is a low-level viremia in patients that we consider traditionally to be successfully treated on HART. This persistent viremia basically can arise from two potential origins. First of which, latently infected reservoirs, and then secondly, your sanctuary sites. So now the question arises, if viral replication is ongoing in the background, then surely we can expect that there's some viral evolution that's going to occur as a result of this. But there's a lot of conflicting results from studies, and they are quite complicated because of extensive analyses of the viral sequences that you need in order to prove this. And also it's been established that treatment-naive individuals that start suppressive therapy and they maintain their levels of RNA levels between or less than 50 copies per mil will actually not have any drug-resistant mutations that will emerge. So if the replication then continues during suppressive therapy, can the low level of viremia be reduced by adding another antiretroviral agent? But it's been shown that the level of persistent viremia is not related to the actual treatment regimen, and that the pre-treatment viral RNA levels, on the other hand, are important, and therefore all regimens can actually potentially inhibit viral replication, and the pre-treatment viral set point will actually correlate with the number of long-lived HIV infected cells. Okay. So what is a reservoir? Okay, so a reservoir is a place where you keep something in store or an extra supply, but the definition for an HIV reservoir is a cell type or an anatomical site where replication-competent forms of the virus will persist for a longer time than the main pool of actively replicating virus. And I would like to propose that basically your latently infected reservoirs can be considered to be HIV's immunity idol. And it basically provides HIV with immunity from being voted off the island of the human body by HART. So keeping this in mind, the latently infected reservoirs are the ones that are sort of linked to the third and the fourth phases of this viral decay. And there's probably more than one class of long-lived HIV-producing cells, but your resting memory T cells are really the class of cells that are the most important contributor, it seems. We know that early on in infection, your HIV will primarily infect the activated CD4 T cells, and it will almost always produce death of the cells. But some of these activated CD4 T cells will be infected while they are transitioning to the resting memory T cell state. And if viral integration has already occurred by this stage, this will then actually cause a latent infection of such a cell. The population of these latently infected memory T cells can also possibly undergo expansion if mitosis does occur and if the cell cycle is entered without induction of viral replication. Now, these infected memory T cells can really persist for many years. And if it receives a stimulatory signal that activates the cell, it can actually induce viral production. So during HART, these cells decay very slowly, an average half-life of about 44 months. And they estimate that under the current treatment regimens, it will take 60 years to actually deplete this reservoir. So I think that's pretty impossible at this stage, it seems. But okay, we'll get to that a bit later on. Now, this reservoir of resting memory CD4 T cells is actually established during the primary infection of the patient, okay? And these latently infected cells do not produce detectable levels of virions in the absence of these activating stimuli. So, in other words, they're not recognized by the immune system or by antiretroviral compounds. Just to mention that there are different phases or a continuum of latency, so to speak, in these cells. And you can see that you get cells that have integrated HIV DNA, but they don't actually transcribe any viral RNA. And then you also get cells that actually do transcribe the RNA, but they don't actually translate the proteins. And you also get some of the cells that will actually produce proteins, but not infectious virions. And ultimately, you get to a cell that is not in the latent phase anymore, and they actually actively produce virions. Integrated HIV DNA can be found in different subsets of memory T cells. Just to mention, most of them is in the central memory T cells and also in the transitional memory T cells. And these basically express specific immune activation markers and proliferation markers. Now, there's two ways by which these infected resting memory T cells are actually maintained. Well, the central memory T cells undergo low levels of proliferation, so they actually just persist for many years. While your transitional memory T cells actually persist by homeostatic proliferation. So it's a very stable reservoir, and this is primarily mediated by interleukin-7. Just some of the characteristics to bring to your attention about these resting memory T cells. They do not express any activation markers, so you will not find CD69 and CD25 on these. They are usually in the G01A stage of the cell, which means that they are sort of stable and not undergoing further mitosis at that stage. And also they express limited levels of the transcription factors NF-AT and NF-kappa B. And also there's very limited levels of or pools of deoxynucleotides present in these cells, which is actually important for expression of the virus. And this is probably the most important point here, that latently infected resting CD4 T cells are in all aspects, apart from the actual integrated DNA genome, exactly the same than uninfected cells. So the body cannot actually distinguish between infected and uninfected cells. So there are some other cells that possibly can also be responsible for viremia during this fourth phase of decay. There are studies that have found sequences that have found that these sequences of the persistent viral populations couldn't actually be found or were rarely found in this resting population of CD4 T cells. So all is not quite known yet. And this can possibly be explained by integration of the viral genome into other cells that have proliferative capacity, for example, stem cells in the monocyte or the macrophage lineage. Essentially, if an infected cell proliferates, it will copy the whole viral genome, and hopefully without introducing errors, and this will generate then an expanded set of these progeny cells to release virus. So what are the important consequences of these reservoirs? Firstly, they create a source of virus that is treatment resistant. And secondly, they create archives of drug-resistant virus that can actually persist for the lifetime of the patient. Which brings me then to the sanctuary sites. Okay, so sanctuary is basically a consecrated place or a place of refuge and protection. And for HIV, it's pretty much the same. And it's an anatomical compartment where HIV replication can continue in the presence of heart. And this is either due to poor penetration of antiretrovirals into that space, or it can be due to properties of that compartment itself, for example, in immunoprivileged sites. So I would like to propose that the sanctuary site is HIV's exile island, because it simply cannot be reached by heart. Okay, so we know that the lymphocytes in circulation are only about 2%, and for the rest of the lymphocytes, they reside in other places, including lymph nodes, the spleen, the gut-associated lymphoid tissue, etc. And this is actually where the majority of viral replication will occur in a patient who is not being treated. In the gut-associated lymphoid tissue, there have been studies that have shown that the GALT more frequently is infected than the peripheral blood mononuclear cells. And also they have found that cross-infection between these compartments do occur during HART, which also is suggestive of possible ongoing replication in the gut-associated lymphoid tissues. Also, the central nervous system. This HIV, we know, can penetrate via the migration of the infected monocytes and macrophages. And if you have an untreated patient, they have found that there are signs of compartmentalization in the CSF and in the plasma. And this suggests the presence of independently replicating viral populations. It's up to date still remains uncertain whether CNS is in fact a sanctuary site, because they have also found that some patients that are on HART and do have a plasma HIV viral load less than 50 copies per mol will also have a CSF level of less than 50 copies per mol. So this is still uncertain. And then the genitourinary tract, of course. There has been studies showing that viral compartmentalization between the genital secretions and the plasma has been demonstrated in some untreated patients. And also there's some further proof, namely that HIV can in some patients be found in the semen, even though the patient is on HART. But the actual contribution of these different possible sanctuary sites and whether they actually do contribute to the persistent viremia is still an uncertain topic, and still a lot of studies will have to be done in this regard. Okay, so having looked at all those aspects, what are the mechanisms of HIV latency? In other words, how is HIV playing this game of survivor? Well, firstly, the main source of latent HIV, as we've said, is most likely this pool of resting memory T cells. And we know that there's two different forms of latency in these cells. Firstly, pre-integration latency, which is not as important. And the reason is, well, basically what happens is the HIV will fuse with non-dividing resting lymphocytes. And basically this viral replication will be blocked before integration actually occurs. So you have basically HIV DNA present in the cytoplasm of the cell, and the half-life of this unintegrated DNA is quite short, so only about a day. So it probably does not really contribute a lot to persistent viremia in HART. But the post-integration latency is what we're really interested in. Here, your infected CD4 T lymphocytes will revert to the resting memory state, as we've explained. And this means that the integrated DNA is extremely stable, and the actual cells themselves are quite stable. And it is basically necessary for the persistence of HIV during HART. Again, during HART, they don't produce detectable levels of virions, except if the cells are activated. Okay, so how does this post-integration latency, how is it maintained and how is it established? So there's different ways, and I'm quickly going to run through each of these different mechanisms that HIV basically uses. Firstly, the viral integration sites. Okay, so the possible position into which this virion integrates into the chromosome can actually affect the rate of transcription. Now, there have been conflicting reports about the actual position into which it does integrate. Jordan et al. Found that latent provirus probably integrates into or close to repetitive DNA elements in the heterochromatin, while Han et al. In 2004 showed that the HIV-1 genomes in the resting CD4 T cells preferentially reside within actively transcribed genes, which could be linked to this phenomenon where you have transcriptional interference, basically just meaning that you have an upstream promoter and that will suppress the downstream promoter. Then you get to the so-called chromatin environment and histone deacetylation, which all sounds very complicated, but it's not that complicated. Basically what happens is if your viral LTR is basically integrated into the chromosome of the host, it can be transcriptionally silent if it's packed into a so-called heterochromatin structure, which basically just means that you've got these little nucleosomes that cover the recognition elements. And as a result of that, it can't actually be transcribed. However, if you get histone acetylation occurring, the nucleosomes will actually be remodeled, and this will make the DNA available again so that your transcription factors can actually bind to the DNA. And this is primarily regulated by histone acetylase and deacetylase, and basically these histone deacetylases actually play quite a critical role in post-integration latency. Then also another method is DNA methylation, which basically just says that high levels of this CpG methylation can actually prevent binding of the transcriptional machinery to the DNA. And basically it will also furthermore form complexes with the histone deacetylases, which sort of further just augments the whole process. Also, the third method is with a lack of key transcription factors. So here, several transcriptional factors we know play quite an important role in HIV transcription, including nuclear factor kappa B and NF-AT, etc. But we're basically just going to focus on the NF-kappa B for now. What happens with NF-kappa B? Basically, the active form will be sequestered in the cytoplasm by an inhibitor. And the inactive form, which is a homodimer, will actually occupy the site at the viral LTR region. Now, this has been shown to also furthermore recruit these HiDACs to the viral promoter, and this furthermore then blocks the binding of the proteins to that particular area. Now, if your resting CD4 T cell is actually activated or stimulated, you get release of the heterodimer or the active form. This will then move into the nucleus of the cell, and this will then displace the homodimer or the inactive form, and this will promote transcription again of the viral genome. So they have shown in studies that low levels of NF-kappa B, of the active form specifically, do correlate with suppressed levels of HIV expression. And then that brings us to the viral activated TAT. We know that this regulates the late phase of transcription and the efficient production of long viral RNA transcripts. And basically what happens is TAT will bind directly to the TAR in the viral LTR, and also it is important for efficient working of the RNA polymerase elongation. And basically it does this by recruiting this PTFB to this particular area, which will then basically prevent all of this from occurring. Stimulate the process to occur. So basically, if it is impaired for some reason, then this will actually promote latency. TAT can also associate with some other important co-activators, so it does seem to play quite an important and an extensive role. And it's also been shown that concentrations and the functionality of the TAT determines expression even more than cellular concentrations of NF-kappa B and all of the other transcription factors. So TAT does seem to play quite an important role. And low concentrations, therefore, or impaired activity will help to maintain the latency of the virus. And then lastly, which is quite an important or an interesting one really, is RNA interference. Basically here, the HIV-1 genome has areas or sequences that actually encode for microRNA. And this RNA interference can actually regulate the gene expression, and this will be by two methods. It can either silence post-transcriptional genes by degradation, or it can also modify the chromatin structure. Okay, so having gone through all of the nitty-gritty of how it's actually doing all of these things, let's look rather at the clinical implications of latency, which ultimately is why we are looking at latency in the first place. So what does HIV's immunity idle actually mean for our game plan? Well, firstly, the latent reservoir is a barrier to cure of HIV. And as mentioned, with the current treatment, it will take over 60 years to actually deplete the reservoir. Secondly, resistant viral quasi-species are actually archived within this latent reservoir. So patients will have lifelong resistance to the drugs that actually cause selective pressure. We know that if we stop HAART, basically the more fit wild-type virus will actually re-emerge from the latent reservoir. As a result, it will become the dominant species in circulation, and the resistant variants are actually stored in the latent reservoir, even though they're not detectable anymore. And basically, if you do scheduled treatment interruptions and you start salvage regimens in patients, your patients, or this will basically not be recommended because these archived drug-resistant viruses will still confer lifelong drug resistance to these drugs. So this is quite an important thing to consider when we are looking at HAART. Then also, if the latently infected resting CD4 T cells undergo activation, then viral gene expression will be upregulated and more virus will be produced. So this can be detected as a low-level viremia, and this actually explains then the transient elevations of viremia, which we call viral blips, which you from time to time will see occurring. Also, just sorry to mention that these viral blips generally are not associated with viral evolution, it seems. Then it also has implications for genotypic and phenotypic drug resistance testing. So we know that genotypic assays will only amplify the most dominant species present at that stage. So you can actually miss mutations that are present in the latent reservoir. Also, your phenotypic assays will determine resistance to single drugs only, and also it will only detect resistance to the dominant species that are present at that stage in the plasma. Also, we know that laboratory cell lines might not always be representative of what's going on in vivo, so this can also complicate matters. So neither of these assays will actually necessarily reflect the resistance of the archived virus that is present in the latent reservoirs. Also then, the development of antiretroviral drugs that have a high barrier of resistance is extremely important and becomes a priority. A high barrier to resistance, as I'm sure most of you know, means that you need a greater number of mutations necessary to actually confer resistance to the particular drug. And we know that this is now becoming a priority, that we try to develop drugs that will not be affected by some of these archived mutations as much as others would. Also then, resistance mutations that are selected by one drug can sometimes infer increased susceptibility to other drugs. So here's a little bit of good news at least. Basically, we know that the M184V mutation, for example, will confer resistance to FTC and to 3TC, but it will also make the virus, the virus basically hyper susceptible to drugs like AZT, especially because the 3TC will maintain selection for the drug-resistant variant. If you, however, discontinue your 3TC, the archived variants that do not have the M184V mutation will emerge from the latent reservoir, and basically you will lose all the effects of the viral fitness and of thymidine analog hypersensitivity. And then the last point is that the latent reservoir can be targeted for future drug development. And this brings me to the last part of our talk, which basically looks at therapeutic approaches to the latent HIV. So how can we outwit, outplay, and outlast HIV? Okay, so there are three broad approaches in an attempt to eradicate latent HIV. Firstly, intensification of antiretroviral regimens so that we can eventually eliminate HIV. But we know that this is disappointing, and the size of the reservoir basically remains unchanged despite us adding more and more antiretrovirals. So this is not really an option. Secondly, we can stimulate the latently infected cells to actually produce virions and to, in other words, make it susceptible to viral toxicity and to immune clearance. And then the third way is that we can stimulate the host immune response to actually clear the latently infected cells itself. Now, the third method has not really been utilized for this purpose yet, and it is a bit unclear as to what extent the latently infected cells will actually produce these HIV proteins. So this is not really something practical that we are looking at yet, although there are some studies going into it as well. So basically, the second point is the one that we're going to focus on. Okay, so we said already that these latently infected cells are in every aspect the same as those that are uninfected. So the attempts to actually eradicate HIV will involve activating these latently infected cells in order for them to express the integrated HIV genome and also then making them targets for immune-mediated killing and for antiretroviral therapy. So in theory, these anti-latency strategies will involve purging this latent reservoir by means of transcriptional activation of the latent virus. And these infected cells will hopefully undergo apoptosis after virus expression, and then HART will then prevent new infections of further new cells. So remembering now that I've mentioned that it's not only the resting memory T cells that we are concerned with, and there can be other cells that are also playing a role in latent HIV. But it might be possible that if we eradicate latently infected CD4 T cells, that this could be enough to prevent rebound after stopping HART. And we know that in this case of the patient with a bone marrow transplant who received stem cells from the donor that was homozygous for the CCR5 delta 32 allele, actually provides some evidence for this. Because although he still has CCR5 positive macrophages that were still detectable in this patient, they could not detect any virus still in this patient. And this could also suggest that the macrophages probably do not play such an important role in the viral persistence. So possibly we can maybe eliminate latent HIV by only targeting the latently infected CD4 T cells. Also, and just one other important point is it's important to identify methods that will not cause global T cell activation, which we know will be quite a devastating thing for a patient. So let's look at the three possible strategies that we can use to purge the latently infected cells. Firstly, we can upregulate cellular transcription to induce HIV expression. Secondly, we can try and alter the effects of the non-coding cellular microRNAs, and also we can try and modify the levels of cytokines that we know are associated with regulation of HIV latency. So the first group, or upregulation of the cellular transcription to induce expression, there are four different groups that that could possibly show some promise. Firstly, the HiDAC inhibitors. Secondly, the DNA methylation inhibitors. Thirdly, the use of prostratin. And fourth, hexamethyl bisacetamide. That took some exercise to get that one right. Okay, so let's quickly look at HiDAC inhibition. We know that this, as we mentioned already, produces or promotes latency by means of regulating the genome structure and the transcriptional activity. So if we have a synthetic HiDAC inhibitor, this can actually lead to reactivation in the latently infected cells. And there has been a study, for example, where they looked at co-administration of the HiDAC inhibitors together with valproic acid and HART, but unfortunately this did give mixed results, and probably there's still a lot more work to be done in this field. Then if we look at DNA methylation, which we know also reduces intracellular transcriptional activity, there are methylation inhibitors, such as this one, which I'm not even going to try and pronounce, Basically, it's been shown in vitro to induce HIV gene expression. It has been found, however, that the DNA methylation probably does not play as an important role in maintenance of HIV latency. So far, the results have not been as hopeful as good as they hoped for. Then prostratin. Prostratin is known to activate NF-kappa B pathways, and it will actually increase binding of the active NF-kappa B to the HIV LTR. And it has been shown to upregulate HIV expression in studies where they used actual mononuclear cells from patients that are on HAART. Unfortunately, it does cause global T-cell activation, and for this reason, it's not a very practical drug and associated with some toxicity in patients. Another possibly better option would be a transcription factor EST1, which has been shown to reactivate latent HIV in the resting memory T cells from patients on HAART without actual general T cell activation. So this is maybe one area to keep an eye on and watch. And then the hexamethyl biacetamide, which is a hybrid bipolar compound that's been tested in oncology trials. And this has been shown to recruit cellular kinases to the viral LTR, and this basically will induce efficient TAT-independent HIV expression. So that's another possible compound to keep an eye on and see what comes out from clinical trials. Then also we can try and alter the effects of the non-coding cellular microRNAs. Here, basically, we know that cellular microRNA is present in a larger amount in the resting T cells. And they basically bind to the 3' end of the HIV mRNA, and this will actually block HIV expression and therefore promote latency. So if we inhibit them, there should be increased protein translation. And so if we try and manipulate these cellular microRNAs, and there are some inhibitors that have been found that basically can do so. This is one possible option then that we can use to try and purge or target the latent HIV reservoirs. And this would be without any T-cell activation, which would also then be a very good thing. All right, then the third way is then by modifying the levels of cytokines that we know are associated with regulation of HIV latency. So here we know that patients that have been put on interleukin-2 and HAART have fewer resting memory CD4 T cells than those that are on HAART alone. And another recent study showed that interleukin-7 can promote HIV latency in those patients that have low CD4 T cell counts. Interleukin-7 is basically a mediator of your T transitional cell reservoir stability. And we can possibly look at something like anti-interleukin-7 therapy as one possible way of actually targeting the reservoir. So in conclusion, we know from ultrasensitive assays that there is a low-level residual viremia that exists in patients that are on HAART with viral copies of less than 50 copies per mole on the commercial assays. And this can possibly be as a result of activation of your latently infected memory CD4 T cells and possibly other cell types which actually make up this viral reservoir. The viral reservoir has massive implications for the clinical management of HIV. For example, archiving of the resistant quasi-species, and therefore we don't recommend treatment interruptions. It has implications for genotypic and phenotypic drug resistance testing. It also may cause viral blips that you could find during routine monitoring of a patient and cause a bit of confusion in the clinical management of the patient. If we can activate these latently infected cells and viral expression can occur, theoretically it would be possible then to eradicate HIV, because we would make it vulnerable then to immune-mediated killing and also to antiretroviral therapy to actually prevent further new cells from being infected. And researchers are attempting to basically outwit HIV by purging its immunity idol, which is our viral reservoir. So the question is, will Tribal Council ever smother HIV's torch, or will HIV be the sole survivor? Thanks. And a special word of thanks just to CBS and Mark Burnett Productions for using their logos, etc. Thank you.